Okay, great. I'm going to go ahead and get started then. My parents are Sally and James McCullough. Good afternoon. We're going to go ahead and get started. Faculty members are going to be trickling in. That's okay. I'm used to this. I'm here today to talk to you about the research that I do, but before I did that, I wanted to advertise UAA STEM Day. We'll be here tomorrow. You saw those tables setting up in the atrium. That's for UAA STEM Day. For those of you who have kids, or for those of you who just want to be somebody that you know excited about science, there'll be hands-on activities, planetarium shows, and lab tours. These are all free and open to the public. So tell your friends, tell your family. It'll be fun. What I'm really here today to do is to tell you about the research that we're doing in my relatively loved young lab. I've been here, I should reintroduce myself. I'm Dr. Catherine K. Milligan-Myhre. You can call me Dr. Cat. It's just a little easier. I'm an assistant professor here at UAA, and I'm starting my 4th year, so this is technically my 4th year review talk. So what I'm going to do is tell you about the research that we're doing. I'm going to focus on using an evolutionary model organism to examine host-microbe interactions, or, as you can also say, microbes in the host, what do fish have to do with it? We do use fish as a model, and I'll talk about that in just a minute. Before I get started, I want to say Fiona to the Denari and Athabaskan, whose land we're on. So what do we study in our lab? We study microbes. I'm actually a microbiologist at heart. That's what I was trained in. And I spent the first 15 years of my life studying these microbes, the ones that make us sick and cause pathogenesis, are potentially pathogenic. And this is typically how people have thought about microbes for about the first 200 years of microbiology, when the first time microbes were observed under the microscope. We spend a lot of that time trying to figure out how to conquer microbes, making antibiotics, controlling their environment, figuring out how they replicate so we could stop that replication. Come on in. But in the last decade or so, we've come to appreciate more as scientists that microbes are actually an important part of us. They help us develop. They stimulate our brains. They stimulate our immune system. They play an important role in keeping us happy and healthy. A big part of studying these beneficial microbes, or microbiota, has been to just figure out who's there. So what I'm showing here is somebody who's trying to write down every member of the microbiota in the gut. And that's because we focus on the gut. So I wanted to highlight the fact that they are looking at the gut. These communities are really complex. There's not just 1 or 2 species. There are actually hundreds of different species in the gut, and they all have very distinct roles. So for the last for about the last decade, most of what we've been doing is just figuring out who's there. Are they always there when people are healthy? Are they, do they change over time, etc.? But really, what we are starting to focus on now, and what we focus on in our lab, in addition to just figuring out who's there, is figuring out how they're interacting with us. So here I have a beautiful drawing of a gut that I did during a seminar like this one. These down here are immune cells that are coming through this gut, coming in between these tight functions to get a taste of what's in the lumen. That's this part. That's the, if you think of the gut as being a tube, this is all the stuff that's on the inside of that tube. And what you can see here is that there's a mucus layer, and there are microbes. Some of them are bacteria, some of them are viruses, some of them are eukaryotes. But there's many different types of microbes there. Now those microbes are playing an important role in the gut. In addition to the things that I talked about, helping stimulate our development and our immune system, they're also doing things like digesting food that we can't digest, and then turning that food into something that we can use, like vitamin K. In addition, they're stimulating the immune system so that if a pathogen comes in, the immune system will be primed and ready to go to attack those pathogens. Now this relationship that the host has, that's us, with our microbes is a really complex one, and it is driven in part by the host genetic background. These are genes that are involved in Crohn's disease and ulcerative colitis, and genes that are found in almost all forms of irritable bowel syndrome. And I'm not showing these for you to memorize them. I'm showing them to show the complexity of these diseases. Now, what all these diseases have in common is that there is an increase in inflammatory cells in the gut for all of these diseases, and the microbiota is disrupted. Now, we don't know exactly which genes are responsible for disrupting the microbiota, if that's what happens first, or recruiting the inflammatory immune response, if that's what happens first. And we're not even sure what comes first, the inflammation or the disruption of the microbial community. All we know is that all of these work together, the host genetic background, the immune system, and these complex microbial communities. Now, studies that have focused on the inflammatory state of the host, so which immune genes are present in the gut and how abundant they are, and microbial diversity, microbial community, including who's there and how diverse they are. These studies have been going on for well over 50 years. Way back in the day, these are images from Life magazine from the 1950s, showing some of the early types of gnotobiotic studies that were being done. Noto meaning known, bio meaning life. So these were studies of which microbes were present and what they were doing. Early on, people would sterilize, have mammals that were giving birth in these disinfectant baths, or they would have entire rooms that were devoid of microbes, and then they would observe what happens to these animals when they are raised in the absence of microbes. And we still do that today. One of the reasons I wanted to show this image was both the complexity of these studies. We don't do the whole rooms anymore. But also the fact that back when these studies first started, they were using animals, and it's hard to tell from this, but some of these were animals with genetic variation. In the last couple of decades, we've really moved away from using animals that have genetic variation to using animals that are inbred. So they, one family line is crossed with members from the same family line to make sure that all the genes are the same. But when you do that, you lose the ability to figure out which genes are involved in this relationship. And as I showed from those images earlier, the number of genes that are involved with this host-microbe interaction are really complex. It's not just one or two genes, there's hundreds of genes, and it's not this gene plus this gene. It could be 5 different genes involved or 20 different genes involved. If you were going to do these studies with inbred mice lines, for example, you would have to do hundreds of crosses to get genetic diversity in your lines. We don't want to do that. So instead, we use this model organism that already has a lot of natural genetic variation called 3-spine stickleback. And we also play around with, a little bit with 9-spine, but I'm going to focus on 3-spine today. The big question in our lab is, how does a host genomic background contribute to host microbe interactions? Because we're interested in how the genes are involved in all of this, we want a population of animals that has genetic diversity, both within the population and between populations. And that's what we have in our 3-spine stickleback. These little fish are found throughout the northern hemisphere. There are many populations within here in Alaska, and these have been used as an evolutionary model for over 100 years. And the reason they've been used as an evolutionary model is because we have those ancestral fish, which are anadromous, meaning they spend part of their time out in the ocean, and then they come up into freshwater to lay their eggs. The juveniles then swim back out into the ocean. So these anadromous fish are thought to be the ancestors of all the populations of stickleback. But occasionally these anadromous fish come up to lay their eggs and then get stuck in these freshwater bodies. And then they develop new phenotypes as they adapt to this new environment. Often they lose their bony plates. So you can see here this anadromous fish has a lot of bony plates. This one has very little. Their pelvic spines get smaller. Their head shape changes. There are many different phenotypes. The phenotype that we're interested in is the relationship between the host and their microbes. as they get caught in these different freshwater populations, these different bodies of water, they're also evolving in these bodies of water that have very different environments than the anadromous fish. These bodies of water have different microbes, different chemicals, different predator, different prey, and we think that because they have all of these differences, that the microbial communities in the gut of these animals will be evolving as well. Now, I like studying in Alaska, partly because here, partly because I love Alaska, but also because Alaska has hundreds of populations, and we have a lot just within driving distance. So here we are in Anchorage, and I'm going to show you some of the sites that we sampled from here in Anchorage. But if you look at a map that has a nice contrast between blue and brown, you'll see that within driving distance of Anchorage, all of these little blue spots are lakes, and almost all of these lakes have stickleback. So that means that if we want to study these different populations, all we have to do is hop in our car and go and grab fish. Or ask our grad students to do that. Now, there are many other benefits for using stickleback. One is that they have very large clutches. And what that means is if you squeeze the eggs out of the female, you can get somewhere between 100 and 200 eggs. And that means if we're going to do a study where we're comparing different treatments, we can have a large number of fish in those studies, and that's a high replicate number. That's great for stats. The other thing is that these juveniles are optically transparent, and actually it's the egg stage through the juveniles. So on the top there I have an egg developing. There's hours post-fertilization after we've mixed the eggs and the sperm. And you can see the fish developing within that chorion. These were not specially treated. This is an actual live image, or not live, but this is a regular image through a light microscope of the fish developing within an egg. And for those of you who haven't stared at these long enough, this is the fish developing here. Here you can see at 96 hours it has some eyes, which are more fully developed. If you zoom in here at 5 days, you would see a nice spine. And here at 8 to 9 days, it hatches. This is still transparent at 8 to 9 days, and in fact, we can take that out a couple of weeks and still be able to see through the fish. The evolution and development has been thoroughly described for these fish. They share many pathways involved in host microbe interactions and immune response with mammals, and in fact, they've been used as a model organism to identify genes that are involved in bone regulation that have been found to also regulate bone development in humans as well. So for example, that bone loss that I was talking about in freshwater compared to the anadromous is due to a gain of a mutation in the gain of an enhancer here, and that same loss of an enhancer led to a change in feet as we were evolving from chimps. And finally, these populations have natural genetic variation. Unlike other model organisms like mice and zebrafish that have been inbred in the lab for a long time, our fish have a lot of genetic variation, so we can see which genes are involved in these different things that we're looking at. Okay, finally, we have a lot of tools to help us examine host microbe interactions. We can isolate RNA and DNA. We can do transcriptome analysis. We can use DNA to sequence the DNA of the host or to sequence the DNA of the microbes that are associated with that host. We can isolate individual microbes on rich agar plates. And so there I have a plate of microbes that were collected from the gut of a stickleback, and we can collect many different types of microbes. I'm going to talk more about that. We can quantify the immune response to microbes. When I was a postdoc, we did this by counting neutrophils, but my postdoc, Lucas, has been working on and has a flow cytometry assay going with help from some folks at the University of Texas. And then we have behavioral and development assays that I'm going to go into here in just a minute. So we have many different ways to study how microbes, which microbes are present, and how they're affecting the host. Now, as I said, a lot of studies recently, in the last 10 years, have really been focusing just on who's present in the gut. And so that's what we're doing as well, because as we establish stickleback as a model organism for host microbe interactions, we need to know which microbes are there. So one of the things that we do, and before I go on, down here you'll see names. The first name is the person who's leading the project. So in this case, Ryan Lucas, my master's student, is leading this project. And he has help from my lab tech, Katie, undergraduate, and now a postdoc volunteer Patrice, and former postdoc Emily. So one of the things that we're interested in is how does the host genetic background select for different microbes in the environment. So the first thing we're doing is just going out and collecting samples, both from the water and from the fish, and determining which microbes are present. Now, for those of you who aren't microbiologists, most bacteria are not the size that I'm showing here. This is not trying to scale. But I wanted you to see the diversity of the microbes that are associated with these fish. What Ryan's doing is he's comparing the microbes in the water, the microbes in the fish. Because we know that that hosts select for different microbes from their environment. And so we hypothesize that the microbes in the water will actually be more diverse than the microbes in the gut. We also hypothesize that different populations of fish will have different microbiota from one population to the other, and that they'll be selecting different microbes from the water because they've been evolving in the presence of different microbes. So he's doing this in a series of field studies. And just so you know, when you look at water, you see that. When I look at water, I see this. He sampled from a number of different sites. So I asked him to put together a nice map that shows each of the sites that he sampled from. And you can see those pins here. Some of them are right here in Anchorage, including Westchester Lagoon. And then we also have some freshwater samples, Big Lake, Bear Paw. And then we have some anadromous populations, Rabbit Slough and Mud Lake. I'm going to point out this Westchester again. It's actually interesting because it has both freshwater and anadromous fish in the same body of water. So it's going to be fun to see if they are selecting for the same microbes or different microbes, as those freshwater fish have already selected for different phenotypes. The X's in this table up here under each population show which years he's been able to go and sample. And so, for example, we've collected from a couple of these populations annually since the lab opened, including Big Lake, which is a freshwater population, and Westchester Lagoon, another freshwater population. And we expanded this year to include, this year and last year, to include Rabbit Slough, which is a population that has been used in a lot of different studies and most likely is the ancestral population for most of these other fish. In addition to collecting from these sites, we've collected from from a series of other sites. I'm not going to have time to talk about Talkeetna Lake chain, but if you want to talk to me about that, Brandon Briggs and I have been collaborating to compare the microbes in the water to the microbes in the gut of those fish as well. And then I also wanted to point out that we do an annual, a monthly collection from Cheney Lake. Pretty much the entire lab is involved in this. And what we're looking at there is the evolution of anadromous to freshwater populations, because we know in Cheney Lake when stickleback were introduced to that lake, and we know which population they came from. They came from Rabbit Slough. We know how long they've been in there. They've been in there for 10 years now. And we also know that they're undergoing a rapid evolution from the anadromous population and phenotype to the freshwater phenotype. So we're following that in the longitudinal study. Longitudinal just meaning we're taking monthly samples to see how the microbiota is changing as these fish are evolving. How do we do this? We go out to these field sites. We throw in these traps. The fish get caught in these traps. We toss back anything that doesn't look like a stickleback. And then we can dissect the guts in the field, or we can take them back to our research lab. In the research lab, we can isolate the DNA from these guts and then sequence them using our MiSeq that Brandon and I own. Or we can take these guts, homogenize them, and collect live bacteria. So for the samples that Ryan is working with, we've been doing a lot of troubleshooting. So Ryan has figured out, optimized the DNA isolation protocol and how to sequence these, and we're hoping to have a lot of sequences coming from this very soon. But we don't have that right now. But what we have done is we've gone out to some of our populations, and we just did a really quick sampling a couple of years ago where we took 10 fish and we sequenced their guts. So each one of these is an individual fish. Each one of these columns is an individual fish. Each color represents a different type of microbe that was present in that fish. So from looking at these, we noticed a couple of things. One is that there's a different microbiota in each fish, and that's inter-individual differentiation and diversity. And we expected that. That's observed in stickleback, it's observed in humans, it's observed in chimpanzees and panda bears and everything else that's ever been sequenced. So that was really cool. We were excited to see that. The reason that we did this initial study is because we wanted to recapitulate the microbiota from the wild populations in the laboratory. Because if we're studying these stickleback, we don't want to bring the stickleback in from the wild into the lab and have a completely artificial microbiota that we're studying in these fish. So we wanted to have a wild microbiota established in the lab. So we took these adults, we squeezed out the eggs, we fertilized them with sperm from the same adults, and we raised them in the lab, and then did the same sequencing on some of those fish. These juveniles were 28 days post-fertilization, and we saw that their microbiota looked an awful lot like the microbiota of the fish from the wild. This was really neat. We saw the same inter-individual variation. We even saw spikes in some of these archaea. We have one huge archaea spike here. We had the same thing in another fish raised in our lab. So we recapitulated this phenotype. It's one thing to look at pretty colors of bars and say that that's the same, that the wild fish look like the juvenile fish. It's another thing to run the stats. So we did that. We looked at alpha and beta diversity from these samples, and we see that the microbiota of our lab-raised fish look like the microbiota of our wild fish. So that's actually really exciting. I'm just going to stop for a minute and say I'm so excited about this. Most people don't have a wild microbiota in the lab. They have whatever floats down from the air in their water. So I'm pretty excited about this. That was led by our former postdoc, Emily Leskack. The other thing that we gleaned from, the other information we gleaned from this is that stickleback microbiota share a lot of microbes with human microbiota. So each of these blue bars indicates that this particular microbe type is found in the stickleback guts or the human guts, and we share a lot of microbes, we humans share a lot of microbes in stickleback. The other interesting thing here is that stickleback share some microbes with humans that aren't found in the other model organisms used to study post-microbe interactions, or two of the post-microbe interaction model zebrafish and mice. So that was exciting. Now, in addition to doing this heavy bioinformatic work and just sequencing microbes, it's not enough to know that their DNA is there. The DNA would be there, and the fish or the microbes might not actually be alive. So to figure out if these microbes were actually alive and to have our own little library of microbes, we started isolating individual microbe types from these guts. We now have a library of over 300 microbes. Some are facultative or obligate aerobic, which means they require air to survive. But we also have obligate anaerobic microbes. This is exciting because the gut is an anaerobic environment. The posterior part of the gut, so the latter, the second half of the gut, is an anaerobic environment. So if we're going to be playing with microbes and we want to introduce them to fish, we want to be able to grab anaerobic microbes because that's what we're going to see most of the microbes in the, that are going to be anaerobic. So to do this, Rachel Cramp is an undergrad in our lab. She worked with Ruth Eisenberg to get this huge thing called the anaerobic chamber up and running, and now she can independently isolate anaerobic microbes from the gut. And in fact, all of these pictures are microbes that Rachel has isolated. And she's gone through and identified a bunch of these, including some facultative anaerobic microbes and some obligate anaerobic microbes. I'm not going to show all of the information here, but we have over 28 different, we have over 25 different genuses now of microbes in our library of over 300 microbes. Now, most of those we identified by sequencing this gene called the 16S gene that's commonly found, that's found in all bacteria and archaea. They have, hey, Skype is now activated. Sorry. They have, the 16S gene has a common area that's found in all of these microbes that we can make primers to, and then they have a region that has slight nucleotide differences, and we can use that to identify one microbe from another. That 16S sequencing allowed us to identify over 280 of our microbes, but there were 20 or so that we couldn't identify by sequencing, indicating that they are most likely have never been characterized before. So, Jessica Kennard, a former undergraduate who's now a term here at UAA, has been teaching, has started characterizing all of these microbes. And the reason for that is if we know what type of microbe we're working with and we can identify it by 16S, we can go in the literature and we can figure out, oh, this is a modal microbe that prefers lactose as a sugar source, and it can ferment it and turn it into an acid. And that gives us some sort of idea of what that particular microbe is doing in these complex communities. But if we're working with a microbe that nobody's ever worked with before, we don't know what that microbe does. And so Jessica has been characterizing the metabolism, so that's what I'm showing in this particular assay. This is a triple sugar iron auger assay to figure out which sugars these microbes can grow on and whether they can precipitate this auger. Also, it's just pretty, so I like this picture. But it gives you a lot of information in one quick assay. And then we have things like the starch assay. Starch is part of plants, and if we have microbes that can break down plants, that gives us an idea that it's involved in the metabolism in the gut. We have phytoglycolate assays to determine whether these microbes require oxygen to grow. And then I'm not going to go through all of these, but we have many different assays that Jessica has used to characterize the biochemistry, motility, and other features like gram-positive versus gram-negative of these microbes. I'm only showing a handful here. She's actually done all of these. And she's now teaching Sharon and Kathleen Sword how to do these assays as well so that we can characterize other microbes as we pull them out of the gut. Now I'm showing this in part because these are completely unknown microbes. Nobody's ever characterized them before. Nobody's ever described them in the literature. We're doing that now. And this is a table showing, this will be one of the figures that we put in the paper that Jessica and I are writing now. And again, Jessica started as an undergrad in my lab. Remember that. But this is a table showing the different microbes that she's characterized and the information that we've learned about these as she's been going along. She's also been able to characterize or identify the genus that's most likely related to these microbes. So if there's a question mark here, they all have question marks. That means this is most likely related to Shewanella, which is a microbe that's characterized, and so we can compare this microbe to known Shewanella and see if it has the same phenotypes. All right. So what else can we do with these microbes besides know a little something about their metabolism? Well, the other thing we can do is we can add them to individual, we can add them back to fish. So we have a protocol that I developed as a postdoc where we can take the surface of an egg that's been fertilized in vitro, so outside of the host. We soak those eggs in bleach and PBPI and then rinse the heck out of them to make sure we don't have any bleach or PBPI being taken over. And then we can grow them in the absence of microbes, so germ-free. Or we can add back complex communities of microbes, those are called conventional, with hundreds of different types of microbes. Or we can add back those individual microbes that we isolated, that's monoassociation. And again, these are drawn to scale. And we can do something fun. We can make mock communities where we only take a select number of microbes from those communities and add them back to the fish. And this is something that Kenny Sparks in my lab has been doing. We can raise fish to 14 days post-fertilization germ-free. This is what those fish look like. Again, they're transparent. We can make out a lot of their developmental markers like the swim bladder, the eye. We can figure out how the spine is developing, how long it is, all that fun kind of stuff. And we do this in a hood. So here we are making our fish germ-free, and then we can raise them in these flasks that allow oxygen to come in, that prevents microbes from coming in. Now Rachel, who is isolating anaerobic microbes, was interested in how these microbes are behaving in live fish. And so she wanted to determine whether microbes that are anaerobic colonize one particular part of the gut or another. You can see on the top there that the gut of the stickleback goes from the mouth, the digestive tract goes from the mouth. There's a stomach, an actual stomach here that looks an awful lot like human stomachs, and that it has gastric pits and is acidic. And then there's, and then here's the length of the gut. And we call this part the anterior part of the gut because it's closest to the mouth, and this is the posterior part of the gut. And so she was, she had this hypothesis that the anterior part of the gut would allow anaerobic microbes, obligate anaerobic microbes to go, because she hypothesized that it's going to be more anaerobic than the front part of the gut. So she took some of these aerobic microbes that we have and put them into the gut, and she cut the gut in half and plated the anterior part or the posterior part. Allowed these microbes to grow on the plate for a couple of days and then counted them. So just from this, you can see that the facultative anaerobic or obligate, but the anaerobic microbes can grow, were more abundant in the anterior part of the gut than the posterior part of the gut. She also took some of her obligate anaerobes and put those into fish. It's hard to see here, but these little dots are the obligate anaerobes, and you can see that they're more abundant in the posterior part of the gut. And then she quantified them, she went through and counted. This is a representation of 3 fish from one class, and this is what she saw in all the classes she looked at, that in 3 fish you would have one that would have a lot of microbes in the anterior part of the gut and very few in the posterior part of the gut. And then the 2 other fish would not be heavily colonized. So this is something that we're working with. This assay, we think, needs to have a higher inoculum. And so we're going to be repeating this. But her initial findings are that facultative microbes are present in higher abundance in the anterior part of the gut compared to the posterior part of the gut. The other thing that Rachel was interested in doing is taking these microbes with a fluorescent protein so that we can make fluorescent poop. And what I mean by that is we can add these fluorescent microbes to our fish and see where they colonize in the live, in a live fish. And when you anesthetize fish, they poop, and so the microbes come out. And that means that you can quantify the microbes along the gut and as they come out. This is using a protocol developed by Travis Wiles, who's a postdoc at the University of Oregon, one of my colleagues when I was there. And several people in the lab are actually making these fluorescent microbes. Katie has taught Sharon Baptista and Kathleen Sardigan, and now Rachel Cramp how to make these fluorescent microbes in the lab. Now, Kat and Sharon have fluorescently tagged, I believe it's 6 strains. Now we're repeating those because we do this at a different temperature that our microbes grow. But they're having success again this semester, so I'm really excited. Okay, so we've been figuring out who is there, both by sequencing and by plating, and we've started playing with what the microbes are doing in the gut. What we're also interested in is what happens to these fish when you disrupt the microbiota, and specifically, does the host genetic background contribute to the ability of the microbiota to help the fish develop? And does it protect the microbiota from being disrupted? So we're doing this in a few different ways. One is doing the extreme, removing all the microbes and raising them germ-free. The second is to add conventional microbiota, complex microbiota, And then adding environmental contaminants. I'm only going to tell you about one perchlorate today. We're also doing a second environmental exposure, contaminant exposure with fluoridone, which is used to treat ammonia, to kill ammonia in Alaska waterways. We're doing that with cat toxin, but I don't have time to show that one today. And then we're also treating fish with antibiotics and seeing how the fish develop in the presence of these antibiotics. Okay. So, if we disrupt the microbiota, how can we tell whether or not the fish has a disrupted development? We can do this by taking lots of different measurements. Some are somatic, and that's just taking a look at this transparent fish and making measurements like swim bladder size, eye size, standard length, which is the tip of the nose to the tip of the tail. We can also look at the behavior of these fish. Emily Luskak and several other students have made a ton of behavior measurements. And then we can look at their gut microbiota, look at their diversity, look at the membership, see which microbes survive in the gut. So first I'm going to tell you about the conventional versus germ-free fish that we've been working with a lot. We took an anadromous population and a freshwater population and made them germ-free. And then we took a bunch of different measurements. What we saw for the anadromous population is that the swim bladder area changed slightly in our fish, but in that the conventional fish had a larger swim bladder than those raised germ-free, indicating that the microbes are affecting the development of the fish. Interestingly, the standard length was not different between the conventional and germ-free fish, and that's actually good because that's what I saw in my postdoc. Now, Emily is really interested in behavior development, and so what she did is she took these flasks that have these fish in them, and you can see them right here, they're tiny, and then recorded them for 5 minutes and looked at their exploratory behavior in the flask, just how much did they move. So, Emily and a large number of undergrads, I'm just showing a couple of here who've done the bulk of the work, Caitlin Miller and Keelan Plank, have done a bunch of measurements where they watch videos of fish and follow the number of squares that these fish move into, so how far are they moving. And they're also counting how long these individual fish spend on the parameter versus the center, and that gives us an idea of how timid they are, because other studies have shown that fish that spend time on the parameter are more timid compared to the ones who are hanging out in the center. And then how many unique squares they go to. So is this fish going here and then here and then here and only going back and forth between these two, or is this fish moving all the way over here? If it's just hanging out in a couple of squares, it's not very exploratory, but if it's moving through a large number of squares, then it's more exploratory. So they've done a lot of measurements, all visually, all manually, and have compared the exploratory behavior of the germ-free and conventional fish. For rabbit slid, what they saw is that if you take all of those behaviors and analyze them together, the conventional fish are different than the germ-free fish. And if you plot this on a PCOA plot, which just takes all of those measurements and groups them by treatment, so conventional versus germ-free, we see that PC1, principal component 1, explains this behavior difference between the conventional and the germ-free. And then if you analyze what's in PC1, you discover that it's mostly that the germ-free fish spend more time on the parameter than the conventional fish. We can do some stats and find out that that's significant. Now we did this with our bootleg fish as well, but there weren't really that many populations, and so I'm not really going to comment on that here. We're repeating it in our freshwater population. The other thing that we measured is nearest neighbor distance. So that's literally figuring out how close these fish are to one another. If they're further away, they're not spending a lot of time with fish. If they're closer to each other, they're spending more time in the general area. And this is a reflection of shoaling, how much time these fish spend together. They did this at 6 different time points. Again, these are 5-minute videos, so they randomly pick time points to determine how close these fish are to other fish. And they did this in 2 different experiments and found that the rabbit slough, which are an anadromous population, those germ-free fish shoal less than conventionally reared fish. So they did not spend as much time together. And in this particular case, the blue bars are the conventional fish and the green bars are the germ-free fish. So, I'm not going to show you all the data from these experiments, but in general what we found is that the freshwater fish, the germ-free, are more exploratory than the conventional fish. For our anadromous fish, there were a large number of differences between our conventional and germ-free. The germ-free decreased exploratory behavior, had smaller swim bladders and a smaller size. In some of the experiments, not all. But both of these, the germ-free spent more time on the perimeter, had decreased shoaling. And for both of these, there was no effect on survival. So even if you remove all the microbes, these fish are able to survive out to 14 days post-fertilization. Okay, so that's our germ-free versus conventional. I'm going to talk about 2 other types of ways that we're interrupting the microbiota. One is using perchlorate. Perchlorate is a contaminant found in many different waterways throughout the United States. We're not showing very many spots here, but there are a bunch of spots here in Alaska as well. It's this molecule that's up here in the corner with the chlorine in the middle. Perchlorate has been shown to disrupt the sodium iodide symporter in thyroid cells. So what this symporter does is it's a protein that allows sodium and iodide to come into thyroid cells. That in and of itself is important, but it's really important because those 2 things contribute to making T4. And T4 is a hormone that's involved in developing fetuses. When you disrupt the ability to make T4, you see dysregulation of metabolism rate, heart digestive functions are interrupted, muscle control is disrupted, the brain doesn't develop as well, and the maintenance of bones decreases. And so what perchlorate does is it disrupts the symporter, and that prevents sodium and iodide from coming in. So then you don't make this T4, and you have all of these different disruptions in the development of the host. Now, Frank von Hippel, who was here at UAA and he's now at NAU, Northern Arizona University, has identified many of these different phenotypes in stickleback and described them in stickleback. To get these, to reverse this, you should just be able to add an excess of iodide because that would allow T4 to continue to be made. But what he found is that when he added iodide to his perchlorate-treated fish, that treatment did not resolve all of the physiological effects that he saw. And so he was looking for another mechanism for this particular chemical. So Emily and I had this library of microbes. So we screened through a bunch of these microbes to determine whether or not they, perchlorate, induced their growth, suppressed their growth, or resulted in no change. And so we did this in our 96-well plate assay, and we grew them up over 22 hours. And what we saw is that if we grew these microbes in control conditions with no perchlorate, or with a salt control, because perchlorate is a salt, that these microbes were able to grow just fine. But when we added perchlorate, some of these microbes had a dramatic decrease in their ability to grow. We screened through a bunch of different microbes, 26 strains. 12 of them had reduced growth. 14 of them had induced growth, so they grew better in the presence of perchlorate. And 8 strains had no effect. These are all stickleback gut isolates. So that indicates that if you threw perchlorate into these fish, that the microbiota would change dramatically in the presence of perchlorate. So we decided to do that. We got some funding from INRI last year for a pilot study. And we looked at both the short-term exposure in adults, hypothesizing that perchlorate changes the gut microbiome in adult stickleback, and a long-term exposure in juveniles for adulthood. This, we applied for a proposal and we didn't get it. But, yeah, we're still trying it. And our hypothesis was that perchlorate exposure induces inflammation in the gut and in adults and delays the development of the immune system in stickleback. I'm only going to show you the experimental design for the adults. So we did adults from Cheney Lake, that rapidly evolving population, acclimated it for a week. We exposed these fish either to perchlorate or instant ocean, which is a salt control because perchlorate is a salt, or a control which had no perchlorate or extra instant ocean added. Did this in triplicate, and then we determined sex, parasite load. There was no difference between the treatments. We looked at parasite load because these are wild populations. And then we looked at the water for in vitro microbiome effects. So how does this affect the microbiota in the water? And then we also looked at the gut DNA for 16S sequencing. This, I'll tell you right now, we're still waiting on. And we did inputs for sex determination. We had a high school student, Elsa Hoffenworth, working with us this summer, and she, or last spring, and she found that the microbiota in the water, in perchlorate-treated water, was significantly less abundant than the control or the instant ocean. So we know that the water microbiota is decreased in the perchlorate-treated fish. We anticipate that the gut microbiota will be as well. We don't know because we don't have our 16S data yet. And then the last thing I'm going to tell you about is an antibiotic exposure. So Emily earned an NSF grant, won an NSF grant to determine how chronic exposure to low levels of antibiotics affects the development of stickleback from different populations. This would mimic levels that we see in wild fish habitats. So this is really getting at the question of how does runoff from the antibiotics that we take affect the development of fish in the wild. And then Ryan Lucas earned an inbreed to look at short-term clinically relevant levels of antibiotics. These levels are similar to what kids get early in development. And the hypothesis here is that fish from different genetic backgrounds would have a different rates of development when they're exposed to these antibiotics. We took fish from 3 different populations, 2 freshwater, 1 anadromous population. We raised them in our little flasks, and we exposed them to ethanol, which is a solvent for the antibiotic we were looking at, which is tetracycline. And then we used low concentrations or high concentrations of tetracycline. We did this for 14 days and in triplicate. At 14 days, what we found is that the anadromous population developed, here I'm just showing standard length, but we measured all the standard development measures that I showed you earlier, had the same standard length whether they were raised under control conditions with ethanol or the 2 concentrations, low or high of antibiotics. And then we looked at our freshwater population, the first one. We saw some effect of the ethanol, and we also saw that the high antibiotics changed the growth of affected the standard length as well. So what was really interesting is that in our 3rd population, which is our 2nd freshwater population, we saw a dramatic effect of antibiotics on the standard length, indicating that the host genetic background affects the ability of the fish to develop normally in the presence of antibiotics. We saw similar patterns for eye diameter, and this is swim bladder area, which has some wild results, which I can discuss with you later. We also saw no influence on behavior or survival, which is interesting. We expected behavior to be different. Now, this is with a really complex microbial community. We wanted to control for which microbes were present. And so Ken Sparks in the lab made mock communities from microbes isolated from Oregon populations of stickleback or Westchester Lagoon here in Anchorage populations of stickleback. And this shows the susceptibility in vitro for these individual microbes. We added these to fish and then added some antibiotics and got these really fun results. This is a really complicated chart. I'm going to go through it relatively quickly because we have 8 minutes left. But I just wanted to break it down this way. So here we have our conventional fish and our germ-free fish. Here we have the fish that were treated with the Westchester Lagoon microbes And this is just a reminder of which microbes they are. And these are the Oregon microbes that we're adding. Now these are all the same population of fish from Westchester Lagoon. So here we're adding Westchester microbes to Westchester fish, and here we're adding Oregon microbes to Westchester fish. We saw that there was no difference in the standard length, or very few, not statistically significant difference in the standard length between our conventional and germ-free, which again, we usually see this. But in our fish that were given the Oregon strains, these were considerably smaller than the strains that were given the Westchester, the fish that were given the Westchester strains. This is just a reminder that they do have different microbes associated in these two different mock communities, and these are what those microbes are. We also saw that these treated fish that were treated with sulfonamide and fish that were treated with tetracycline to disrupt their microbiota had different, the sulfa treated fish were considerably smaller than the fish that were not treated or that were treated with tetracycline. And these were in the Westchester Lagoon fish, Westchester micro-treated fish. Interestingly, the microbes that were disrupted with sulfonamide looked really similar to the Oregon population. And so one of the things that I've asked Kenny to do is go through and look at these different microbes and figure out how different these Bacillus, Aeromonas, Pantonia, and Pseudomonas look from the microbes that we see in this population. And then in the fish that were given the Oregon strains, what we saw is that the fish that were treated with tetracycline were larger than the fish that were not treated with antibiotics. And the fish that were treated with sulfonamides was in between. So this is fun. We're trying to interpret this based on the different microbes that are present and working on writing this up right now. In our future directions, we have so much sequencing to do. Those of you who have ever been in my office or who have walked by my office have seen this chart. These are all the samples that we need to isolate DNA from, make libraries and sequence, analyze, and write up. There's not much here. There's a whole lot here. So we have DNA isolated, we just need to sequence it and analyze it now. And then these are all the different papers that we're working on. We have 11 papers we're hoping to get out of the lab in the next 2 years. There are a lot of projects I didn't get time to talk about, and I'm really sad. One is the kombucha one, where we're isolating microbes from kombucha and trying to figure out if they can kill Helicobacter. Jeremiah Lewis is doing this in undergrad in my lab. I love this project. And then we have this really great pathogen exposure experiment that I've been wanting to do for about 5 years, and that Lucas finally did this summer with our med student Anastasia. In this one, we're looking at whether or not the host genetic background allows microbiota to protect against pathogens. We're just starting to get DNA, or just starting to get results back from this, so I couldn't really discuss it here today. But if you want to talk to me about pathogens, I studied them for 25 years, I'll talk to you all about it. I have a huge lab. I've been really lucky to have amazing people work in my lab these 3 years. I've had 3 great postdocs. I have 2 graduate students who just started this fall. Kelly and Kenny were both undergraduates who decided to stay on to do their master's. Ryan has been a master's student with me for 2 years. He's writing up stuff now. I had a medical student from Wyoming working with me over the summer. I've had amazing technicians who do everything that I don't have time to do. I've had post-bac volunteers. Tyler's shown there, but Jessica and Patrice are both post-bac volunteers as well. Means they've gotten their bachelor's degree and now they're just hanging out in the lab because they're awesome. These are all the undergrads with me. The folks in blue have moved on and they're doing fantastic things. The folks in white are still with me. I've gotten funding from INBRI, the American Fisheries Society, NSF, and NIH. NSF and NIH. I have fabulous collaborators who helped me with all of these projects, and we occasionally get together and take weird pictures. With that, I have 3 minutes to take questions. Why not? Hey Brandon, what's up?
For your isolates, did you sequence the full 6 units?
Yeah. If we're identifying, we sequence the full 16 sequence.
Have you been able to resolve it even at the phyla level? Or?
Yeah, we're down to the genus and species level, so we can identify individual species, and then we're hoping to do, we're going to that nanopore day in November, so we can figure out if we can do whole genome analysis on these guys. We've already gotten 8 of them sequenced fully. I had a colleague at the University of Oregon who did it for us for cheap. We just need time and a person to analyze that data. Yeah,
have you ever looked at the array of microbes from a given batch of eggs versus from different fish?
I want to do that. So we do all of our processes in vitro. So it's in a petri dish with sterile gloves and in the laboratory and whatnot. But what I would love to do is go out into the wild and grab some clutches of eggs. There's a very small time point. There's only 8 days between the time that egg is laid and fertilized and the time that the fish emerge from those eggs. But I think it would be really fun to find out if there are any microbes that protect the eggs while they're sitting at the bottom of the lake. We know that for other fish and from squid, that there's this protective goo that hangs out over the eggs, and that there are microbes associated with those that prevent things like fungal outbreaks from killing the eggs. So I think that would be really fun. We haven't done it in our lab.
But in that diagram where you have on the right-hand side the juveniles, and I think each bar up and down represented from one fish, right?
Yeah.
Were those all from different batches of eggs from different?
Nope, there was one batch of eggs, so we took one clutch of.
So there you are looking at the variation then within the eggs from the same genetic background.
Yeah, so that's exactly what we're doing there. And now at this point, they're not eggs anymore; they're juveniles. But we do see individual variation between one juvenile and another, just like we see with adults, and just like folks have seen with zebrafish or others. But it would be fun to do at the egg level. The eggs are, when you squeeze a female and she lays her eggs, they're in a clutch that's covered with this vitelline stuff that holds the eggs together. So it'd be difficult to individually pull out each egg and look at the microbiota of them individually because they're covered with that goop. But it would be fun to do that. So we look at them at the juvenile stage when their microbiota has been established. Their guts opened, they're taking in water from the environment and food, and the microbiota should be established at that point. Yes, hi Don.
Hello. It's amazing to me that your notobiotic fish attain the same size that the inoculated fish do.
Yeah.
And it kind of indicates that gut microbiota doesn't really do them any good.
Well, let's think about this.
Trials at about 14 days. Is that part of the perhaps reason for that?
Yeah, so 2 things. One, we're only looking at 14 days. So that's incredibly early in development. These fish can actually live out to 2 years, maybe even 3 years. But we're just looking at the first 2 weeks. The second thing is other researchers have raised notobiotic mice all the way through entire life cycle. So creating a new generation in these notobiotic bubbles. And so we know that from other animals, you can raise fish or raise animals in the absence of microbes, and they develop fine. What they don't do is they're not as good at digesting their food, so you have to change the percentage of protein and other things in their food. And their immune system is not stimulated very well. And so if you give them a pathogen, they die much faster than an animal that's been raised in the presence of microbiota. Yeah?
With your behavioral protocol, it seems like you could very easily quantify sleep, although it would take 24 hours of watching this probably. And sleep influences the immune system, growth hormone, and which might influence length? Have you ever thought about looking at such?
So what we've done is we've controlled the light in the incubator that these fish are raised in, and that could control sleep patterns. Other studies have looked at zebrafish, and the sleep is really wonky in these fish. It's not like they have a real circadian rhythm of sleeping for 10 hours and awake for whatever. They tend to sleep in short little bursts. And I'm not completely sure that it's light dependent. I know there's another researcher who's looked really heavily into behavior, Allison Bell, who Emily trained with, and another couple of folks who've looked at behavior. And I don't know if they've looked at light specifically, but that would be something fun to look at. Emily and I, in one of her first proposals, actually proposed, it was one of the internal proposals, proposed raising these fish in different life cycles and looking at what happens to development, behavior, and immune responses. Didn't get funded, so we didn't do it. All right, go enjoy the 105A. Go enjoy the food and the drinks for soda that's available in 105A. And thank you.